Here we go. Oh, let me get that shut. There you go, buddy. Okay. Let me get this started up here. What happened? Okay. I've got a delay going on here, so I'm gonna be a little off on this. Okay. As you read this, let me know if you can hear me or not. It looks like everything should be good on my end. Let me get this straightened out. Good grief. Okay. And I just did this a few minutes ago. Should have been more ready. Okay. Sorry about this. Alright. Yeah, so my kid was sick today and I had to turn around as she threw up in the car to come back, and so I'm not able to drop them off at daycare, so we'll do this online, which, it's not that bad. Okay. So here we go. What we're going to talk about today is photosynthesis. And I normally like to really do this live because I really, really enjoy going through all the light reaction live rather than doing this on a PowerPoint. But hey, this is what we have with us here. So let's take the big picture here of photosynthesis, right? I mean, let me make sure I got it here. Yeah. So the big picture, photosynthesis. Photo means light, synth means putting together. With photosynthesis, we're going to put together by a light. So what we're doing is we're going to take some carbon dioxide, some water, and energy. Those are the reactants. And we're going to use that energy of sunlight, and we're going to make glucose, carbohydrates, and release oxygen in the process. And this is an endergonic reaction because the products, glucose specifically, has more energy than carbon dioxide or water. It's got much more stored energy in its bonds. And basically, so what this photosynthesis is going to do is it's going to reduce the inorganic carbon and carbon dioxide to an organic form. So we're going to add some electrons and hydrogens to it. And thanks for letting me know you can hear. So basically, by reducing this carbon into the carbohydrates, we need a source of electrons, we need a source of energy, and we need a source of hydrogens. So what photosynthesis is going to do is going to use, some specialized pigments, some molecular structures, some cellular structures or organelle structures to basically convert energy and sunlight and store it as a carbohydrate by fixing carbon dioxide. Now, basically, just to remind you, this is a redox reaction, reduction oxidation. So when you're reducing something, you're gaining electrons. So we're going to reduce the carbon and carbon dioxide to a carbohydrate by adding electrons to it, and along the way some hydrogens as well. And then what's going to happen now is we're going to take water. That's going to serve as our source of electrons and hydrogen. So basically, we're going to use some enzymes and energy to strip the electrons away from oxygen. Get this right. We are going to oxidize oxygen. That's pretty cool, isn't it? Okay. It's telling me I'm not getting enough video to maintain smooth streaming. Oh boy. Nice pause on that one, wasn't it? Okay. So, what's going on here? Okay. Sorry about the pauses here. I can see myself pause there for a second. But basically, we're going to oxidize oxygen. And it's going to be released as O2. And water is going to serve as that source of electrons. And remember, this is the endergonic reaction here. The endergonic reaction is the products, glucose and oxygen. They have more potential energy than the reactants. So let's take a look at this overview of photosynthesis. We've got to take this energy and sunlight, and we're going to capture that energy. And we're going to ultimately transform that energy into ATP. This is the energy currency of life. And we're going to use that ATP, and then we're going to strip the electrons and hydrogens from water, and then we're going to ultimately use that in what's called the Calvin cycle. And in the Calvin cycle, we can reduce the carbohydrates. So the light reaction is going to capture light energy, transform it to ATP, give us our source of electrons. And the hydrogens there. So, all right. So interesting. Remember this, that the light reaction and the Calvin cycle are linked by this flow of electrons. So the light reaction, the photo part, okay, I mean, we have an idea what's going to happen. We've already seen a lot of this before. Basically, we're going to split water. I've already said that. It's going to release, this is where the O2 comes from. The oxygen doesn't come from carbon dioxide, it comes from water. And those electrons, since they're coming from water, they don't have a lot of energy. Okay? And because they don't have a lot of energy, well, the photosystems are going to elevate those electrons in energy. So they're going to capture the electromagnetic energy and light and use that to elevate electrons so that they have energy so we can do work with them. And then once the electrons get they're going to be energized, they're going to help make ATP, and they're going to also be carried to the Calvin cycle by an electron carrier called NADPH. And we'll talk more about that later. And then the hydrogens, they have an interesting route too. Once they get removed from water, they actually get pumped into the thylakoid membrane, into the thylakoid space, where they're going to be used to make ATP. Where have we seen protons making ATP before? Chemiosmosis, cellular respiration, right? Okay, so if you remember the structure of a chloroplast, it's a double membrane structure, and on the inside of it, you have these flattened sacs that are stacked. And these flattened sacs are called a thylakoid, and they're stacked into what's called a granum. And basically, the light reaction is going to occur on the thylakoid. So, we're going to get some light coming in, and we're going to use that light to basically split water with some enzymes, and elevate electrons and energy. And basically, we're going to ultimately make ATP. That's going to be where the energy in the sunlight has been transformed and stored as ATP, which is the energy currency of life. And then the electrons will then be used to reduce an electron carrier, NADP, to NADPH. And the H on the the H on the NADP is, of course, coming from partly water as well. And remember, NADP is very similar to NAD and NADH that we see in cellular respiration, except the one with the phosphorus group on it is a very strong molecule, basically used in a lot of anabolic reactions, especially where reductions are needed. So it's used not just in photosynthesis. Okay, now the synthesis part of photosynthesis is the Calvin cycle. And the Calvin cycle occurs out in the stroma. Okay, and basically what it's going to do is it's going to take the electrons from NADPH and the ATP, and it's going to take carbon dioxide and it's going to fix it into an organic compound. It actually doesn't make glucose. That's actually made after the Calvin cycle. But it's a metabolic process. It's a series of chemical reactions. Okay, and those chemical reactions are a series of reduction reactions that are, like I said, we're going to add electrons and hydrogen to the carbon atoms. Okay, so there's the overview of photosynthesis. And importantly, the light reaction is connected to the Calvin cycle by this flow of electrons through NADPH. Okay? All right. So back during the COVID, I made these slides with a lot of words on them so that, you had all the information. And I've just never taken those words out, so I apologize if these slides have lots of words. They're also posted in the module folder as well for week 2, but they give you just a lot of information. I feel like it just not want to take out all this information. So the light reaction, what it's doing, and you can read this, it's taking electromagnetic energy, right? This is a form of kinetic energy in the photons and their wavelengths that they're moving in. and it's going to transform that kinetic energy into chemical energy, ATP. And yeah, let's take a look here. So the electrons, when you split them away from water, they have very little energy. So we've got these photosystems, all right? And these photosystems are going to elevate these electrons in energy. Let's take a look at it here. So basically, as you pull these electrons away, they have such little energy, so the photosystems will capture the energy in sunlight. And if you remember yesterday, I mentioned that those photosystems, they might need a little quantum mechanics to work on them. But for right now, we'll go with the classical explanation that they basically, through inductive resonance, they concentrate the energy into the reaction center, which then elevates those electrons up very high. Okay? Now, there are two photosystems. Photosystem 2 and photosystem 1. I know I said 2 first. Photosystem 2 basically was discovered second, but it actually comes first. So once we've captured the energy in the sunlight, we split water, we've taken the electron from the reaction center, we're going to send it high up in energy, and it's going to be picked up by an electron carrier called phaophyton. Normally I like to draw this on a board, but I don't have my whiteboard set up behind me right now. But basically, what this photosystem 2 is going to do is capture this energy. And there's lots of reaction centers, lots of proteins in here, lots of pigments forming all of this structure here. And basically, hold on, I'll get back to your question right here in a second. Once you take that electron and send it up, it's going to be picked up by an electron carrier from photosystem 2 called phaophyton. And then phaophyton is going to take this electron through the thylakoid membrane. So the photosystem is in the thylakoid membrane. So is what you're seeing here, the cytochrome complex. So phaophyton takes the electron from the photosystem 2, and it's going to take it to another thing called the plastocyanin, and we'll take it through the cytochrome complex, and then the other electron carrier will then take it to photosystem 1. So basically these electrons get elevated energy, go through an electron transport chain, and the cytochrome complex is actually used to pump protons into the thylakoid space. And then as they do that, they're doing work. They're going down in energy. So the photosystem 1 will then re-elevate those electrons so they can be carried to the Calvin cycle. Now, Violet, I see that. Does the Calvin cycle and light reaction happen in the thylakoid? And the answer is no. Only the light reaction occurs in the thylakoid, and the Calvin cycle occurs in the stroma of a chloroplast. The stroma, of course, being from the membrane of the thylakoid to the inner membrane of the chloroplast. So it's that empty space out there that doesn't have the thylakoids. Okay. You know, these reaction centers, we're still figuring out not just the reaction center, but also the entire photosystems, figuring out exactly where all the different proteins are that forming the protein complexes, learning where almost every single atom of these things are located. But this is a very large molecule. Basically, you know, they're made up of proteins and they're made up of lipids, pigments like chlorophyll and carotenoids, and other cofactors. So these are really large molecular compounds here. And that P680 that you see in the middle, and that you also saw back here in the reaction center, that P680 tells you that this reaction center does really good at picking up wavelengths at 680 nanometers. Okay, now, these are the photosystems. I mean, large molecular complexes is pretty cool. You know, 20 subunits, 99 cofactors. I got to read this. I can't remember all of it. And like I said, all kinds of electron acceptors and everything else. And it's green from all of the chloroplast in there.
Hey, Daddy, can we have the ice?
Go ask your mom for some ice, okay?
Daddy, Mommy's gone.
Okay, I'm busy, so I'll have to get you ice when I'm done, okay? Okay. My kid's coming in here asking for ice cream. Okay, I'll get you some ice in a minute, okay? Okay. Or Lily, get it and then hand it to me. Okay, So, thanks for understanding. Like I said, I've got kids. Remarkably, when you're going to school, they feel horrible. Then you turn around and come back, and all of a sudden they miraculously feel better. But my kid did throw up, so there is that threshold that I'm like, yeah, you can't go. Okay. So as we go through the light reaction, remember we've got these two photosystems, photosystem 2, photosystem 1. There's a cytochrome complex. There's an ATP synthase, which actually makes ATP through chemiosmosis. And what we want to do now is, I've already kind of talked about what's happening with the photosystems elevating the electrons, but we're going to follow these electrons from water through the photosystem, and we're going to see how they capture that light energy, make ATP, and reduce NADPH.
I already got this ice.
We got some ice cream here. I always take the dad tax. There you go. The dad tax. That's what they have to pay for having me open their ice cream for them all the time. But they don't seem to mind too much. Okay, so let's get back to this. What we're going to do is we're going to follow these electrons. So we're going to see how this light reaction ends up reducing NADP to NADPH, the source of electrons for the Calvin cycle, and makes ATP, which of course is the energy needed for the endergonic reaction. To that text. Okay. All right. So, of course, the electrons, as you know, they come from water. They get elevated by light in the reaction center. Then as they get elevated, they get picked up by a molecule called phaophytin. And phaophytin will then take it to another electron carrier called plastoquinone, which then takes it to the cytochrome complex. Now, this cytochrome complex, this is another really large complex. That tax.
Thank you.
It's another really large complex, protein complex you can see here. And in fact, all those little ribbons in there, those are the beta pleated sheets, and all the different colors represent different subunits, of this large protein complex. It's big, right? I mean, it's huge. And what this is going to do ultimately is take the electrons from our electron carrier. So remember, water, reaction center elevates them. They go up. Then they go picked up by phaophytin, the plastoquinone, to the cytochrome complex. The cytochrome complex is a proton pump. It's going to pump protons from the stroma into the thylakoid space. So it's building up a proton gradient. Excuse me. My kids are not the only one getting sick. I'm coming down with something too. Okay. Of course, my pups have to come in and say hi to me now as well. So at any rate, the cytochrome complex, this is a proton pump. And like I said, we're going to start creating a proton gradient. We're storing potential energy inside the thylakoid space. So we're lowering the pH. We're making it more acidic inside the thylakoid space. And that's interesting because we've seen exactly this in cellular respiration in the mitochondria, where the electron transport chain, right, is a series of proton pumps that pumps protons into the inner space of the mitochondria, creating a proton gradient. This is doing basically the exact same thing. In fact, it was probably evolved from that in the begin with. So the electrons that were elevated in energy by light, now that energy, the electrons, is being stored as a proton gradient. So now, once we have the electrons are now removed from the cytochrome complex by another electron carrier called plastocyanin. This is plastocyanin. It uses copper. Hence the name cyanin. And it's very blue. And then it will take the electrons to the photosystem 1. Because once those electrons have gone through the electron transport chain, they lose energy. All right? They're losing energy. So the role of photosystem 1 is to, once again, to use the energy in sunlight to elevate those electrons back high in energy again. Okay, so once it does that, all right, now we can take those electrons and they're elevated from photosystem 1. Now there's another really important electron carrier called ferredoxin. And basically, ferredoxin is going to remove it from photosystem 1. And ferredoxin is basically this really interesting iron-sulfur enzyme that's used in a lot of metabolic pathways. And basically, I mean, it may be important for the origins of life. Pretty interesting stuff there. Then the ferredoxin uses a reductase, and it's going to basically take that to the NADP reductase to reduce NADP to NADPH. Now, NADPH is similar to NAD that we see in cellular respiration, but the difference here is it's got that phosphorus group, and instead of used in catabolic reactions like NAD, because cellular respiration is a catabolic reaction, we're breaking down the glucose. Here we're going to make glucose. This being anabolic, we often see NADP, and it's used in many other processes, not just here. In fact, we use NADP as well. Okay, and then that NADP H will then be used in the Calvin cycle. So now we've got this flow of electrons, and you can see the entire chart here, where we start with the splitting of water. We're going to basically split 2 waters, get 4 protons, and an oxygen, an O2 molecule. The photosystem 2 is going to elevate it in energy. Then it's going to be picked up by that phaophytin. That's an electron carrier. And it's going to take it to plastoquinone. And then plastoquinone is going to take the electron to the cytochrome complex, which is our proton pump that will then pump protons into the inner space of the thylakoid, right? And then the electrons will be picked up by plastocyanin, taken to photosystem 1. From photosystem 1, it will be elevated back in energy, will be picked up by ferredoxin. Then ferredoxin will take it to NADP, where the reductase will stick the electrons and the protons onto NADP, forming NADPH, which will then be used in the Calvin cycle. And of course, because we now have a proton gradient, the next thing we have now is that proton gradient is then used to make ATP through chemiosmosis. And once we start making ATP through chemiosmosis, now we've converted light energy into chemical energy of ATP. And we now have our source of electrons attached to our electron carrier to be used in the Calvin cycle. And throughout this whole thing, you've noticed I keep comparing this to cellular respiration, because to me this is really important. Yeah, okay, just picking up the top chat there. But if you notice that there is basically this similarities between the electron transport in mitochondria and photosynthesis. The reason is, all of this evolved 3 billion years ago, and whether or not you're doing cellular respiration or photosynthesis, making ATP through chemiosmosis is pretty similar. You set up a proton gradient with an electron transport chain. So electrons got energy that you're using to pump protons across some membrane to create a proton gradient, which then makes ATP through chemiosmosis. The light reaction and cellular respiration both do the same thing. The differences are, and I like to ask this often as a test question, the big difference occurs is the source of electrons in the light reaction in photosynthesis, of course, is water. The source of electrons in cellular respiration, of course, is organic molecules. The source of energy in photosynthesis, the light reaction, is the sun, because when you pull them off the water, they don't have much energy. And the source of energy in cellular respiration is, the energy is actually coming from the energy stored in the organic molecules and the bonds between the carbon and the carbon and hydrogen. And then the fate of the electrons is different too, In cellular respiration, the electrons end up on water, or oxygen, I should say, to make water. And the fate of the electrons in photosynthesis, the light reaction, goes to the Calvin cycle to reduce organic molecules. So those are some similarities there. So make sure that you know this. Okay, so there's a summary of the light reactions. This is what you end up with. And then make sure that for any tests we have coming up on Thursday, that you know these things here. Like I said, this PowerPoint has already been has already been posted, and I've got some squawking in the background. Hopefully it's not too bad. All right, so that is the light reaction. Now, let's take a look at the Calvin cycle. And this will be the last thing that I do today, is the Calvin cycle. Okay. Let's pull this up, get this going. I'm going to go shut my door again. I would normally shut the door and latch it, but the handle on it doesn't work anymore, and it doesn't have screws to take it off, so I've got to replace basically the whole door. Wonderful. That's what you get when you have cheap doors. Okay, all right, so we've already gone over the big picture here. We know a lot about the big picture of cellular oh, wait, am I on the right one here? Yeah, okay, just making sure I have everything. What we're going to focus on now is we know that we've got this endergonic reaction happening. This is the overall reaction right here, where we're taking our carbon dioxide and our water and we're making a carbohydrate out of it and releasing oxygen in the background. But the reality is this is a multi-step process, and photosynthesis actually doesn't even make glucose. It makes a 3-carbon compound. And of course, if you remember, I've said this over and over, that this is a endergonic reaction here. So, big picture here, Calvin cycle. It's often called the dark reaction, but it doesn't occur in the dark. It has to have light. So the Calvin cycle is dependent, though, on the light reaction, because the light reaction has to be going at all times to supply a source of energy, ATP, and to supply those electrons. Okay, so here we go. You can't have carbon fixation without the energy. This is an endergonic process. So the Calvin cycle has 3 phases. One thing that we're not going to do here is memorize all the chemical reactions, the entire metabolic pathway. We're going to do things a little bit different here. So, here we go. Okay. Oh. We have carbon fixation. This is catalyzed by an enzyme called rubisco. If you remember from the 80s, there's this company called Nabisco. I always think about that. But rubisco stands for ribulose bisphosphate carboxylate oxidase. That's a mouthful, but that's what it stands for. But the name tells you a lot, that there's a ribose sugar with 5 carbons. It's a bisphosphate, so there's a phosphate group on each side. A carboxylase, which means it can add a carboxyl group to something. And if you remember carboxyl group, that's a carbon double bonded to an oxygen and single bonded to an oxygen. CO2. It's like carbon dioxide, right? So that's your carboxylation, is adding carbon dioxide to it. The oxidase, tells you it's an enzyme, is that it can also add oxygen to organic molecules as well. So rubisco, ribulose bisphosphate carboxylase oxidase. It's an enzyme that sticks carboxyl groups or oxygen to a ribulose bisphosphate. The next stage after fixation is reduction. That's where we're going to start reducing those carbons by adding electrons and hydrogens to them. And then I don't remember adding in those things. That's so funny. The regeneration of the carbon dioxide acceptor, RUBP, which is the ribulose bisphosphate oxidase. So here it is. Let's take a look at it. This is the Calvin cycle. It took Benson Calvin almost a decade to figure this out. And the big breakthrough was realizing that this is a cycle that goes round and round and round. That's the third part, is a regeneration. Regenerating RUBP, the ribulose bisphosphate. So if you look at this right here, let's see if I can get a pointer here. Let's see how this pointer works. I'm trying it. Let's do a laser pointer. Okay, so now I think you guys can see that. Input. If you notice, it inputs 3 carbons at a time. Now, for each turn of the cycle, I mean, each one is actually, 1 carbon, but we're going to do 3. And the reason why we're going to do 3 is because the output down here is a 3 carbon sugar. Okay? So we're going to start right there. There's our carbon fixation. We're going to take ribulose bisphosphate, which is this 5 carbon compound right here. It's a ribose sugar. Remember, it's got 5 carbons. 1, 2, 3, 4, and 5. There's our phosphates at each end. Okay, now we're going to take the enzyme Rubisco, and we're going to stick a carbon dioxide onto RUBP. Now, this is a really unstable molecule that lasts for millionths of a second, and it quickly splits into something called 3-phosphoglycerate, or 3-PGA. And the 3 tells you where the phosphate is. And then basically we now have 6 of these, because we're going to start with 3 RUBPs, adding a carbon dioxide to each one. They're going to split, so you're going to get 6 of these things total. Okay, notice ATP to ADP uses energy. Now we're going to go into what's called the reduction phase. Reduction occurs over here. If you notice, there's NADPH that's being reduced to NADP+. It's adding electrons and hydrogens to this. Now we're reducing it. Then down here, we get out 1 3-carbon sugar, 1 G3P sugar. And that's going to be used to make glucose eventually. So that's why we start with the 3 right here. Then the next stage is reduction. Or the regeneration stage. I'm sorry, my kids are squawking in the background. Of course, mom just left, so who knows what's going to happen in the next few minutes. But during the regeneration phase, we've got these 5 3-carbon compounds. And basically, those 5 3-carbon compounds, we have to stick them back together to form 3 5-carbon compounds. So we got 5, 3. Now we're going to go 3 to 5. So we still have the same amount of carbons, but we only have 3 of the RUBPs now. And that's the regeneration. And notice that it does require ATPs again. So this whole process is taking, between, 15 and 18 ATPs for the entire process of the Calvin cycle. And it also requires 6 NADPHs. And that's to get 3 carbon atoms fixed into 3GP. Okay. So I got a couple questions here. So the electrons are at their highest energy levels when they are in the photosystems. That is absolutely correct. You nailed it. That is right, Jasmine. And then Benjamin, what's the difference between 3-phosphoglycerate and glyceraldehyde 3-phosphate? That's a really good question. The biggest difference between the glyceraldehyde 3-phosphate and the 3-PGA is that the glyceraldehyde 3-phosphate, if I were to draw out the molecule, if I was in class, I'd draw the molecules for you. It has more covalent bonds to hydrogen and to carbon. So it is a reduced molecule compared to 3-PGA. And if you look right above it, right over here at this 6 NADPH, it's adding in hydrogens and it's adding in electrons. So not only is this molecule more reduced, the glyceraldehyde 3-phosphate is more reduced than 3-PGA, the 3-phosphoglycerate, it also has more potential energy. And if you think about a sugar molecule, there's basically no double bonds in it anywhere. No carbon or double bonds between carbon or carbon and oxygen, right? It's all hydroxyl groups for the most part, and carbon and hydrogen. Excuse me. So it stores a lot of energy. So that's the big difference. It's more reduced, more hydrogen, more potential energy. Man, those are fantastic questions. All of you guys are asking really good questions. So thank you for doing that, for asking them. Okay. Now, interestingly, we've got this enzyme called Rubisco, and it's once again it stands for ribulose bisphosphate carboxylase oxidase. The ACE tells you it's an enzyme. The RUB or the Rubisco part, that stands for the ribulose bisphosphate, and then it's going to stick either a carboxyl group or an oxygen to it. RUBP evolved very early on in photosynthesis. And in fact, it evolved so early into photosynthesis that there was basically no oxygen in the atmosphere or dissolved in water. So this thing has an affinity for oxygen. So it can actually do what's called photophosphorylation. So when carbon dioxide levels get low inside of a leaf, it can actually fix oxygen to RUBP instead of carbon dioxide. But interestingly, Rubisco is maybe one of the most abundant enzymes on Earth, and probably one of the most common one, or one of the most important ones, because it's the one that's fixing carbon dioxide, and it's occurring in every green plant, and everything that you see that's on a plant that's green basically has this molecule in it, this large enzyme complex, which is pretty cool. Okay, so Rubisco is a pretty interesting enzyme. It's got 16 subunits, 8 active sites in it. It's an ancient molecule, or I should say enzyme's been around for a long time. Now, because Rubisco, when it fixes carbon dioxide to RUBP, it forms 3-PGA. I know, 3-phosphoglycerate. It forms a 3-carbon compound. Hence, this is called a C3 pathway. All right, and so that's called a C3 pathway. There's also a C4 pathway that I'm gonna get to the next lecture. I'm actually I think I'm coming down with something like my kids. I'm getting exhausted sitting here talking to you guys online here. So I'm gonna stop here, and I'm gonna catch up. Or tomorrow, I'm gonna do cam systems and introduce water movement in plants, and then that will be it for the first test. So thanks again. Thanks for everybody coming in, and I will almost certainly see you tomorrow in class. My daughters are already feeling better. You know, she threw up on the way into school, and then I think she's already feeling better. I'll come if I'm feeling bad. All right, I will see you guys later. Y'all have a good one.